Our final talk this morning is by Raj Majeti, also from the Department of Medicine, and Raj got his funding from the Stem Cell Institute.
Good afternoon, everybody. I'm very cognizant that I stand between you and the lunch that I already saw outside when I walked in. So I learned this new little feature of PowerPoint. You can turn the arrow into a laser dot when you have multiple screens. That's my contribution to the learning today. But anyway, I'm Ravindra Majeti. I'm from the Division of Hematology, and I'm going to tell you about a work in progress where we're trying to understand the stem cell effects of germline RUNX1 mutations in a disorder called FPD-AML, familial platelet disorder related to acute myeloid leukemia. And AML, acute myeloid leukemia, is a very aggressive cancer of bone marrow progenitors in the blood. It occurs in both adults and kids. It's more common in adults than it is in kids. But in the genome sequencing era, aligned with previous studies of human genetics, what's been discovered is that there are mutations in the germline that can predispose some individuals and therefore their families to develop myeloid neoplasms, myeloid cancers. And so there's a list here taken from a recent review by one of the leaders in this field, indicating that there are multiple genes that can be mutated, and in these pedigrees for these families, there's an increased risk of developing AML. Sometimes it occurs on its own, other times with platelet disorders, as is mutations in the RUNX1 gene. And then there are other organ dysfunction syndromes that are associated with these various genetic mutations. Now, as it relates to RUNX1, there are heterozygous germline mutations in this gene that cause FPD-AML. And these kids from birth have severe thrombocytopenia in some cases, but they certainly have functional platelet defects, leading them to have bleeding disorders and other related problems. But unfortunately, about 30 to 40, maybe 50%, depending on the mutation and the pedigree of individuals, will progress to develop a myeloid malignancy, most cases AML. And the average age of onset is young, 34 years. AML as a disease has an average age of onset in the broader incidence over the age of 65. And more importantly, these individuals have very poor prognostic disease. They can only be cured with a stem cell transplant, allogeneic stem cell transplant. Now, what is RUNX1? RUNX1 is a transcription factor. It's widely expressed in hematopoietic and osteolineage cells, jumping on the theme that Sean was showing us about the importance of both compartments in blood development. And RUNX1 comes in several different isoforms. They're illustrated here, but the schematic of the protein is here, where there's a RUNT homology domain that's responsible for making protein-protein interactions with another transcription factor called the core binding factor beta subunit. Incidentally, both RUNX1 and CBF-beta are involved in common translocations in garden variety AML, not germline-associated AML. And this transcription factor also has a transactivation domain, and together this complex forms and can transactivate multiple genes involved in various processes of hematopoietic differentiation and regulation. Now, the mutations that occur in the germline disease, the FPD-AML syndrome, many of them are illustrated here on this schematic. There's a variety of missense mutations. There's also nonsense and frame shift mutations that can occur distributed through both the RUNT homology domain and the transactivation domain. And it's a complex set of biology. Some people believe they're loss-of-function mutations. Others are dominant negative. The general sense is that they are all reducing the transactivation ability of the transcription factor. Now, people have been interested in exploring the genomics of how FPD progresses to AML, starting with the germline mutation. In most AML cases in these individuals are associated with loss of the remaining wild-type RUNX1 allele, so it leads to deficiency of RUNX1 activity as part of the transformation process. There are co-occurring mutations in different genetic pathways that are involved in leukemogenesis, BCOR pathway and the cohesin complex with the STAG2 member. And as I alluded to, there's complex penetrance and genetic anticipation that can be observed in the affected families. So penetrance meaning only a portion of the individuals who carry the mutation ever develop the leukemia. And it seems to be that there are families that exhibit genetic anticipation, meaning that each generation the disease is occurring in younger and younger individuals. Now, importantly, the mouse models that have tried to mutate RUNX1 have led to a lot of understanding about RUNX1 biology, but they can recapitulate the platelet defects of the FPD syndrome, but they don't recapitulate progression to AML, and that is a barrier to trying to understand what it is that does lead to progression. And this is a schematic. I know it's small to see, but the concept is that there are secondary mutations that are acquired, leading to some type of clonal expansions that ultimately progress into frank AML. And there may be contributions from inflammatory cytokines and the bone marrow microenvironment, stromal cells, that can contribute to this disease. So we set out to try to model RUNX1 deficiency and RUNX1 FPD mutations in primary human hematopoietic stem and progenitor cells using the CRISPR-Cas9 system. And this illustrated here schematically. We start with CD34 positive cells that are enriched for the stem and progenitor cells, and we introduce into them the RUNX1 guide RNA that will target the RUNX1 gene, along with the Cas9 nuclease that will cut the locus. We're trying to cut it here in exon 3. But in not just wanting to cut the locus, we want to modify that locus. And these double-stranded DNA breaks that can be introduced by the Cas9 protein can be repaired through homology-directed repair. And we use a technique or technology that we learned from the Porteous lab here, where we use a recombinant AAV6 virus that can deliver that template, which consists of a left arm of homology and a right arm of homology. And in intervening sequence, we can introduce a fluorescent tag, in this case a GFP-expressing transgene, so that after that homologous recombination has occurred, the endogenous RUNX1 location is now disrupted, and integrated into that locus is a fluorescent protein that we can use to monitor, isolate, and follow those cells. And in fact, you can use two different viruses, one with a green fluorescent protein, one with a red fluorescent protein, and ultimately then isolate cells that have both fluorescent proteins that have disruption of both alleles. That way you know what the genotype of the cells is that you're working with. So here's a FACS plot showing that it is possible to use that approach that I showed in the cartoon and generate cells that are double positive for the fluorescent protein. And we did a PCR experiment just to confirm that the RUNX1 endogenous allele is actually modified, and that's true. And you can see that the RUNX1 protein is completely deficient. So we took those cells and now looked at their biology, what happened to them when we studied them. First, we did liquid culture assays and either differentiated it into the myeloid, the erythroid, or the megakaryocytic lineage. And we found there was no defect in their ability to make myeloid cells. And the control here are cells that are modified in the exact same way at the AAVS1 locus, which is relatively considered to be a safe harbor within the human genome. But when we differentiated them to megakaryocytes or erythroid cells, the RUNX1 cells had a defect. And that's consistent with the phenotypes that's observed in those individuals. In fact, we did a slightly different version of this assay, where instead of just looking at cells in liquid culture, we looked at them in methylcellulose, a semi-solid media, to look at progenitor function. And again, we saw a lack of erythroid production with the RUNX1 knockout cells, as well as a lack of megakaryocyte production. Now, we also wanted to look at their ability to proliferate and to retain immaturity, meaning expression of the CD34 marker. And when we put these in culture conditions, we found that the RUNX1 knockout cells proliferated less readily and retained their CD34 expression longer than the controls, suggesting that they may have a propensity to retain some of their stem cell properties. We also took these cells through the editing process and then transplanted them into immunodeficient mice to look at their ability to recapitulate hematopoiesis in vivo. And as I mentioned, this is work in progress, so small numbers of cells, but we don't see any gross abnormalities in their ability to engraft in immunodeficient mice. And the final experiment we did with these knockout cells was to mix the wild type, or the control ones, with the RUNX1 knockout cells in equal proportions, transplant them in vivo, and look at how they competed for their ability to engraft. And we found that the RUNX1 deficient cells were much more impaired relative to the controls in their ability to engraft. Certainly makes us think a lot about how the second allele gets lost in these individuals and still progresses to leukemia, but certainly trying to work out the phenotypes. Now, the individuals who have this disease, they have heterozygous mutations in the RUNX1 gene. And so we wanted to try to model that by knocking in a RUNX1 cDNA into the endogenous locus that we could then modify exactly what the particular mutation and variant was. And in this approach, we'll end up with one allele that's knocked out and one allele that has the RUNX1 protein expressed from the endogenous promoter. Now, going back to the schematic, there are many different mutations. We certainly can't model them all, but we picked some that were in the transactivation domain as well as the runt homology domain to try to model the different variants. Some of those they're schematized here with the different mutations and what the protein that would be produced from those are. So here we have engineered them so that we could get these RUNX1 variants through homology-directed repair into the 34 positive cells, and you can see the frequency with which these were able to be recombined into the locus. And we've shown here that the integration is not random, it's actually occurring where the Cas9 guide is cutting, and that we're able to get expression of the mutant RUNX1 transcripts with protein expression as we might expect. Here's the RUNX1 knockout, and here's one of these frame nonsense mutations, and here's one of the point mutations which still is producing the protein. So I don't have data yet to show you about what the functional consequences of these variants are. That's the work in progress. But I'll just wrap up by letting you know what we've shown is that we can get efficient introduction and expression of multiple mutant RUNX1 alleles in primary human hematopoietic stem and progenitor cells, and that these RUNX1 knockout cells exhibit defects in the erythroid megakaryocytic lineages, both for differentiation and with competitive engraftment in vivo, and that we can engineer these FPD AML-associated mutations, mutant alleles, into the HSPCs. It's the only way we're going to get cells to really study this. Can't stick a bone marrow needle into those individuals all the time and extract their bone marrow. And our next steps are really to generate these heterozygous variants and assess their functional effects through the assays I just showed you. And we also have a parallel set of experiments. I don't have time to show you the data, but where we're trying to assess the cell non-autonomous effects, looking at RUNX1 mutated MSCs engineered in the same way. And these are very much like those leptin receptor positive cells that Sean talked about this morning, where we can look at the interactions between the niche with RUNX1 deficiency and the hematopoietic cells with RUNX1 deficiency. So our goal is really to establish a model where we can get progression to frank leukemia and ultimately try to understand that process. So with that, I'll stop there, acknowledge funding from the CHRI, and we've also been funded through the FPD AML RUNX1 Research Foundation, which is part of the Alex's Lemonade Stand Foundation. And this work has all been done by a very talented graduate student, Amy Fan. Amy's got a poster right outside if you're interested in more details. So thanks, I'll take some questions.
So at the risk of holding you from lunch for a minute or two, we do have time for a question or two. Please use the microphones, though, if you would.
Ravi, where you're presumably for each experiment, you're redoing the CRISPR modification in a new sample.
That's correct.
And so to what extent do you see, is it an issue having biological differences from sample to sample, either because of differences in the patients or differences in the recombinations?
Yeah, that's a great question, and it relates to the technology. There's definitely difference in the recombination frequency from specimen to specimen. Some of that is probably intrinsic to the individual cord blood donor. Some of that may be related to the logistics of getting cord blood into the lab after the delivery. As it relates to the biology, I think that the general phenotypes are similar across multiple different cord blood specimens, but the magnitude and the efficiency will vary.
Tony.
So Ravi, that was great. So you've disproven the loss of function of the RUNX causes the leukemia. So now if the audience wants to know what's your hot model for leukemia would be a second hit, and how is this related at all to why, like in myelodysplastic syndrome, you get leukemia as well? I mean,
yeah, no, that's a good question. I think that it's very perplexing in many ways that the second RUNX1 allele deficiency leads to this loss of stem cell function in vivo. I think that we need to really work that out a little bit more. But it may be that those cells undergo a bit of a crisis and they acquire additional hits, and that will lead them to progression. That's a genetic hypothesis that we need to prove through manipulation of the cells. But as it relates to MDS, it's a very interesting because RUNX1 is also mutated in sporadic non-familial cases of both MDS and AML, where it is a very bad risk feature, and it's not clear that the mechanisms will be the same in the familial syndromes as it is in the sporadic cases.
Well, Ravi, thank you very much.
Thank you.